everyone. I'm Dr. David Perlmutter. Welcome again to the Empowering Neurologist. On our program today, we're going to talk about cancer. And, truth be known, we're not making a whole lot of progress in dealing with cancer. Cancer rates continue to climb by and large. We're going to be talking to Dr. Thomas Seyfried, who's the author of this incredible book called Cancer as a Metabolic Disease. Dr. Seyfried writes about how cancer may represent a manifestation of changes in what are called the mitochondria, those small energy-producing particles that live within each of our cells. Let me tell you a little bit about Dr. Seyfried. He received his Ph.D. in Genetics and Biochemistry from the University of Illinois. He also holds a master's degree in genetics from Illinois State University. He was a postdoctoral fellow in the Department of Neurology at Yale University School of Medicine, and then served on the faculty as an assistant professor of neurology, again at Yale. Prior to receiving a full professorship, Dr. Seyfried was an associate professor in the Department of Biology at Boston College. He has had a variety of honors and awards over the year from such diverse organizations as the American Oil Chemists Society, the National Institute of Health, the American Society for Neurochemistry, and the Ketogenic Diet Special Interest Group of the American Epilepsy Society. Dr. Seyfried has previously chaired the Scientific Advisory Committee for the National Tay-Sachs and Allied Diseases Association, and he presently serves on several editorial boards of well-respected scientific journals, including Nutrition and Metabolism, Neurochemical Research, and the Journal of Lipid Research. His research program right now focuses on gene-environmental interactions, how our genome interacts with various environmental influences to cause disease and to threaten our health. He has special interest in things like epilepsy, autism, and brain cancer, as well as other neurodegenerative conditions. Dr. Seyfried investigates many of these diseases from the perspective of genetics, but also from the interplay between our genetics and energy metabolism. Much of his work also has direct translational benefit to the clinic, meaning direct applicability for doctors who are actually treating patients. So I'm real excited about today's interview. Let's get started. Well, as I mentioned in the intro, I'm very excited to chat with you today, Dr. Seyfried, and welcome to the program.
Well, thank you very much. It's a pleasure to be here.
Well, I'd like to start off, you've dedicated your life, at least as of late, to looking at things like epilepsy, Tay-Sachs, some neurodegenerative conditions, gangliosidosis events. But I think what's really gotten a lot of people's attention is the contention that you have with reference to the underlying cause and possible way out of this real mess that we seem to be in as it relates to cancer. And again, just for everyone's benefit, there is this, I think, a landmark book that healthcare practitioners need to have in their bookshelves. So first, maybe an overview as to what is the conventional approach today to dealing with cancer.
Well, it hasn't really changed much in the last, maybe 50 years. Basically, there's a diagnosis, a biopsy, if you will, or some image that will indicate something might be wrong. And then based on histological preparation, a decision is made as to whether this might appear to be benign or, say, more aggressive, malignant, if you will. And then a decision is made as to what might be the best course of action to attempt to manage the disease. And, most of the minor cancers can be easily managed with a surgical resection if it's shown that this is not a malignancy. And the case is closed. Okay, so many people who so-called might have considered themselves having cancer basically have a benign lesion of some sort, and it's easily resolved. The issues come when there's some consideration that maybe there's something going on here. The number of mitotic figures, what the cells might look like to the professional pathologist's eye, and then a decision is made. A decision is made to perhaps be a little bit more aggressive. So not only do we have the surgical resection, not always, depending on the kind of tumor it is, but then this could be followed by a series of recommended chemotherapies, followed by either radiation, chemo, sometimes a combination of both. Most of our work has been done in brain cancer, so that's a pretty established procedure. The individual would be presenting with some sort of a neurological problem of one form or another, or a limb that's not working properly. A scan of the brain is shown to have some sort of lesion. Oftentimes, depending on the image that you get from various scanning procedures, a decision is made to either immediately resect this tumor or possibly have some watchful waiting period. The problem, of course, is in going into these tumors, especially in brain, you provoke the microenvironment by the very surgical resection that you do, which often leads to then a dissemination of tumor cells that are, in other words, the bees are out of the hive, the horse is out of the barn. The very act of disturbing the microenvironment can precipitate a more aggressive situation. Yet this is what is generally done. And then this is followed, of course, by a series of radiation and chemo, temozolomide, often in conjunction with dexamethasone, a steroid. Often dexamethasone can be given to other cancer patients to improve their appetite when they're sick from some of the chemo. The latest idea is the immunotherapies, which are more or less just simply an extension of the kind of chemotherapies that are already being used. They seem to focus a little bit more on our immune system's ability to try to kill the tumor cells. But in a sense, they're not much different, either in the in their therapeutic effectiveness or in the kinds of adverse effects they produce. So we're still dealing with a problem, and the problem, in my mind, stems from the fact that the majority of therapies that we use today are based on the idea that cancer is a genetic disease, and these genes cause a destabilization of cell proliferation. So many of the chemotherapies that are used are very toxic to try to stop DNA replication. And this obviously leads to hair loss and a variety of other maladies that are affecting any kind of a proliferating cell in our body, whether it's tumorigenic or not. So we suffer a lot of these indirect adverse effects from the very therapies that are used to try to manage the disease. Now, of course, many of these therapies do, in fact, cause a resolution of the disease, provided it, in most cases, as long as it's not a stage 3 or stage 4 condition, which are the most aggressive. Many of the chemos, radiation, and surgeries that we use are corrective. There's thousands, millions of people that have been treated with cancer that seem to be doing okay. The problem is we've put these people now at risk for a variety of other ailments that they have as the result of being treated for the disease that they have. But there's a new branch of medicine called cancer survivors medicine. So we have a whole range of different neurological, gastrointestinal, cardiovascular problems that occur years, perhaps 10, 20 years after, which then overall lead to other kinds of problems, reducing the quality of life in one way or the other. So we have a problem here. We're not using therapies that are very effective, that can enhance quality of life and improve overall survival. And we know about these therapies. They're being used successfully, but not in mainstream medicine. And that's where we think viewing the disease as a metabolic disease will eliminate a lot of the problems that we are currently experiencing in the cancer field at this time.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the General Scientific Concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So your work is really an outgrowth of the pioneering work of Otto Warburg back in the 1900s, a work for which he ultimately in 1931 was awarded the Nobel Prize. And Dr. Warburg really felt similarly that this was an issue caused by changes in the functionality of mitochondria that ultimately caused these mitochondria to shift their metabolism away, to doing what's called aerobic glycolysis, fermentation, which is not, it's a situation that's associated with increased inflammation, increased free radical production, ultimately further DNA damage of the mitochondria themselves, and probably secondarily damage to the nuclear DNA as well. So this was the work, and you must have seized upon this work and had an epiphany one day. And how did that affect you?
Well, it didn't come when I first started a lot of the work that we were doing. I only heard of Warburg's name, didn't really understand what he had done, because every textbook discusses Warburg's biochemistry. You know, he was the preeminent biochemist of the 20th century.
Right.
And he had done a lot of things. And you hear Warburg's, in glycolysis and lactic acid fermentation and these kinds of things, and respiration. But it didn't connect with us until we started to use, as a matter of fact, we were given a drug, an anti-ganglioside synthesis inhibitor that seemed to work really well in stopping tumor growth. And of course, the company got very excited about this. But then when we did all the appropriate controls, we found this drug was working really well. But when we did the appropriate controls, it turned out that the drug was interfering with the digestion of foods that were eaten. So it was like an indirect calorie restriction. So then when we put in the control experiments with the mice that were fed to match the body weight of the drug-treated animals, the tumor reduction was the same. There was absolutely no difference between. So this opened our eyes to say, whoa, this calorie restriction is very powerful. And we did a lot of studies on this. And then we began to ask, what is the mechanism by which calorie restriction shrinks tumors? And it became clear. They lower blood sugar and they elevate ketones. And this links directly to Warburg's central theory that the cells need glucose. And the question is, how is it possible that these tumor cells need so much glucose? And Warburg's key contribution was showing that the respiratory system of the cell is damaged. And if the cell is going to live, it has to engage alternative sources of energy. And the most primitive source of energy we know on the planet is fermentation energy. And aerobic fermentation, or they say aerobic glycolysis, is simply a reversion back to the situation that existed on the planet for all organisms before oxygen came into the environment. Back then, a couple of 2.5 billion years ago, all the organisms on the planet at that time were fermenters. So this is an heirloom that has now become reinvigorated in these tumor cells. So, and then it became clear that they ferment two major fuels, glucose and glutamine. And glutamine is metabolized to glutamate, which then generates tremendous energy from TCA cycle fermentation. It's a different kind of energy. It's an ancient energy pathway, just like in the cytoplasm is glycolysis. So these tumor cells are simply reverting back to the way all organisms existed on the planet before oxygen. So one has to simply ask, what kept those cells alive, and what did they do? So they fermented. Okay, what did they ferment? They ferment glucose and glutamine. What happens when you deprive tumor cells of glucose and glutamine? they die. So the unique thing that Warburg didn't know, he didn't know substrate level phosphorylation in the TCA cycle, and he didn't know that ketones could not be fermented, or if he did, he didn't write much about it. So what we simply do is prepare patients or mice, and we transition their whole body over to high ketones and low glucose, which is done through ketogenic diets. This prepares the body for the various drugs and procedures that work synergistically with the diet. The diet alone will not resolve cancer in most patients. However, when it is used with specific drugs and procedures, it's unbelievably powerful. And it's only when you put these together that you get this in synergy, which can target and kill tumor cells without toxicity. And this is the nice thing.
You just said something I think that's really very important for our viewers, actually several things. And that is that you're not proposing that this ketogenic approach is going to cure all cancers by any means, but is used along with as an adjunct, sort of in a systems approach. And our viewers have seen that before, as the Dr. Dale Bredesen's approach, for example, to Alzheimer's, that multiple ideas are utilized to create the most favorable environment for dealing with a situation. So again, to simplify then for our viewers, what you're saying is that the cancer cells basically are going to require sugar or they don't have the ability to utilize these fat derivatives, these ketones, as a fuel source, as opposed to our normal non-cancerous cells, which can use these ketones as fuel sources. And you're exploiting that by basically depriving these cancer cells of what they need, i.e. sugar and carbohydrate-derived fuels.
Yes, yes, and it works beautifully. I've seen spectacular results. In fact, it works much better in humans than it does in the mice. We were very excited to see how powerful these approaches were in the animal models, but we have yet to cure a mouse of metastatic cancer or glioblastoma. Some people will say, oh, they cure cancer in mice all the time. Not the mice that have the same kinds of cancers as humans have, I can tell you that.
Well, just for our viewers, I'd like to just recommend that you consider Googling on the Huffington Post, an article I wrote summarizing Dr. Seyfried's work several years ago that dealt with several of your published cases dealing with glioblastoma, a very challenging type of brain cancer, extremely aggressive, but by using, again, this ketogenic approach as an adjunct, as an add-on therapy. And again, our viewers watching this are pretty well clued into other upsides of being more ketogenic and reducing their carbohydrate intake. So thereafter, where do we stand then in terms of mechanistically, what are we doing to these mitochondria, these energy producers, when we are feeding them ketones as opposed to having them shifted to just utilizing glucose or glutamine?
Well, the ketones cause metabolic stress in the mitochondria of the tumor cell, but they enhance the metabolic efficiency in normal cells. So, one of the things when they say cancer cells are drug resistant and radiation resistant, and they have this capability, that resistance in those tumor cells is the result of their fermentation pathways. So both glucose and glutamine increase dramatically glutathione, the antioxidant capability in those tumor cells. So the tumor cells are resistant to many chemotherapeutic drugs and radiation because they're fermenting like crazy. So if you're going to make them vulnerable to these kinds of therapies, you have to remove the fermentation shield. And you do that by gradually removing glucose and gradually removing glutamine, which now puts these tumor cells at incredible vulnerability to a little bit of elevation in oxygen. And the ketones will facilitate that, but so will hyperbaric oxygen. So if you
I was just going to ask you that.
Yeah,
Who knew?
See, hyperbaric oxygen alone, like the diet alone, is not likely to have major therapeutic benefit to patients. All right, but when you prepare the patient metabolically and then use hyperbaric oxygen together with drugs that put a little bit more restriction on glucose and glutamine, the results are unbelievable. I mean, these tumor cells are just decimated, and it works remarkably well in humans. I've seen data in humans now from Egypt and Turkey, where they're less restricted in the way they treat cancer patients, with spectacular results on a variety of stage 4 cancers, using minimal amounts of chemotherapy together with ketogenic diets and hyperbaric oxygen and a few drugs like 2-deoxyglucose and some of these that work together. And the results are almost incomprehensibly good. I don't think once this becomes known, I think it's going to be earth-shattering. It's just that people always ask, where are the clinical trials to do this? Where is the evidence-based medicine to support what we're saying? And the issue is here, of course, is clinical trials are double-blind crossovers to look for a singular drug on a particular problem.
Exactly.
We have to change that because the way we use metabolic therapy is a combination of drugs, diets, and procedures that are dosed and strategically used in scheduling and timing. And all of this varies depending on the individual. So one shoe does not fit all. So this our view of how we manage the disease has to be changed dramatically to incorporate this new way of managing cancer.
I want to just go back for a moment to our discussion on treating patients with this brain cancer, glioblastoma. And in one of your case reports, you mentioned how one of the individuals was treated with steroids, with dexamethasone. And interestingly, one of the things that we as clinicians see commonly when patients are given steroids like Decadron or dexamethasone is their blood sugars go up.
Yeah.
So, in that regard, then, while you're acutely reducing inflammation and you look at the repeat MRI scan and you see, oh, the tumor looks smaller, the degree of inflammation surrounding that tumor looks reduced, we must be making progress. The reality is, based upon your thesis, that giving a drug that may very well increase glucose availability might be absolutely doing more harm than good in the long run.
Yes, well, we've written about that often. And just recently, within the last year, the work from the group from Stoop and colleagues, who actually write the standard of care for glioblastoma worldwide, have now come out with a paper urging caution in using steroids for people with brain cancer, glioblastoma and other cancers, for the very reasons that I've mentioned. It does a lot of damage in other ways to facilitate, actually, it gives you false information about the success. If you have a patient who has a brain tumor and that tumor is growing and it has a lot of edema, it generally will press on some region of the brain, causing some maybe paralysis in the face, the arm, or the leg. And when you administer the dexamethasone, you reduce the edema, and it makes the patient feel like there's a miraculous change in the tumor.
And everybody's happy and feeling confident, but in reality, it's a Faustian maneuver.
Oh, it's terrible, because everybody, oh, look, I'm feeling good now, the various things. But the problem is that you've reduced edema, but you're also increasing the rate at which the tumor cells are growing.
Right.
And, despite the fact that we've been saying this for a number of years, that dexamethasone should not be used against brain cancer patients for the most part. And the recent paper coming out from Stoop's group, who defines the standard of care. You can go into any neuro-oncology clinic today in the top hospitals, and they are using huge doses of dexamethasone to treat their patients. It's unconscionable. I don't know if they don't read the literature, they're unfamiliar with the metabolism. I just don't understand it.
I think it goes right back to the short-term gains, clinical improvement as measured subjectively and objectively by performance. And like you say, people suddenly feel better, they regain function, and everybody's happy. And that is really short-lived, that sort of improvement. And then the back end of the hurricane comes around. I want to go back to the notion, really.
There's one more thing I'd like to say about the standard of care for glioblastoma. And it's not just the using of the dexamethasone and elevating blood sugar. It's also the fact that when you irradiate the brain, you create a tremendous amount of necrosis, which then frees up the glutamine. And the glutamine now becomes a prime fuel. And when you put glucose and glutamine together in the microenvironment, you send up the perfect storm of adverse effects that will lead to the rapid growth of the tumor and the demise of the patient. Everybody knows this is palliative anyway. So it's not the standard of care for glioblastoma is not considered curative, but the problem is so many patients think it is. And it's a real tragedy of monumental proportions to think that we are treating people with procedures that are going to create an environment that's going to lead to the rapid growth and their demise. And then we throw in the toxic drug temozolomide, which is often used together with the radiation. And this creates more necrosis, more problems. And now recent paper showed that the temozolomide increases so-called driver genetic mutations, which make no sense at all. Yet you have a little bit of an increase in overall in progression-free survival, not overall survival. So the whole concept of how we're treating brain cancer is upside down. It's not right, and the results are clearly in the survival statistics, which are abysmal. But those abysmal survival statistics are directly the result of the treatments that we're using. I know so many patients who have abandoned the standard of care, who are using metabolic therapy and are doing so much better. And the problem is, where are the clinical trials for this? Well, who's going to do this?
Yes.
What hospital is going to say, we are going to abandon radiation and chemo and do metabolic therapy?
Well, beyond that, who's going to underwrite those studies?
Yeah, that's another issue.
Yeah, and just to be very clear about it for our viewers, that is that studies are underwritten by people who develop these monotherapies or these drugs in the hopes that they are successful and hence their product will be the standard of care. I want to go back to your contention that this is ultimately, cancer in general is ultimately a mitochondropathy. It is an acquired mitochondropathy, and we've been talking about that in terms of Parkinson's and Alzheimer's for quite some time. But it is an acquired failure or disruption of the way that mitochondria are involved in energy production. That said, first walk us through that, and then secondly, let's talk about this notion of mitochondrial therapy as sort of being the moniker under which your program takes its support.
Yeah, well, first of all, Albert Szent-Györgyi, also a Nobel laureate, back in the 1979, published a very interesting paper in Proceedings of the National Academy of Sciences, describing what he referred to as the oncogenic paradox. The paradox basically said, how is it possible that all these so-called provocative disparate agents from the environment or some rare inherited mutations might produce cancer through a common pathophysiological mechanism? And this was an enigma, as it was, as a paradox. Some people got cancer from being exposed to cigarette smoke. Other people got cancer from being exposed to a chemical carcinogen. Some people got cancer from being exposed to radiation. I mean, it was like many different origins on the surface. But what was common to all of these, and that's what I put together in the book for the first time, was that all of these impact either directly or indirectly on the mitochondria. So you have inherited gene mutations that increase your risk for cancer, but the products of those gene mutations damage the respiration. Every one of the provocative agents in the paradox, in one way or another, will damage the respiration, but not too acutely. It's a chronic, progressive damage to the respiration, and in order to survive, the cell has to upregulate a more primitive form of energy. So what happens is, once the mitochondria in a particular population of cells, in a particular organ, in a given patient, those cells signal, that mitochondria signal with a very sophisticated biochemical mechanism, the nucleus. And the nucleus responds by upregulating genes that turn on fermentation pathways. So if the cell cannot ferment, it can never become a tumor, because all tumor cells are fermenters. So we have certain cells in our bodies, like neurons in the brain. They can't ferment. So when you take away their respiratory capacity, they either die or they shrivel up or they have these problems. So you don't get tumors very often from neurons. You get tumors in the brain from glial cells. And glial cells, or even heart cells, muscle cells. If you have cells with a lot of mitochondria and you damage the respiration, those cells often die. They don't usually become tumors. But on liver, kidney, spleen, bladder, and breast, and lung, those cells have a capacity to ferment when respiration is damaged. So those cells can go on and form tumor. So you have to have cells that when respiration is chronically damaged, the cell has the capacity to upregulate fermentation. And in that process, there's a vicious cycle where mitochondria become more damaged, leading to reactive oxygen species, which then, as you said, damage the nucleus, causing the gene mutations, which are largely red herrings. So the majority of the cancer field is focused on red herrings. They are the effects. They are not the cause of the problem. The cause of the problem is the damage to the respiration, which then leads to a cascading series of events, which most people are focusing on. Oncogenes and tumor suppressor genes. The somatic mutations are simply an effect of ROS damage. Oncogene turn-on are transcription factors that upregulate fermentation. So if the cell is going to survive, it's going to need fermentation. It's inefficient, so you need more glucose transporters, you need more glutamine transporters, you need all the things that cell is going to need to ferment, and that's what the oncogenes are doing. But they're an effect, they're not the cause.
There's a mechanism, at least with respect to neurons, whereby when there are deficiencies of mitochondrial function, compromised energy production, and ultimately changes in the transmembrane electropotential, that through transmembrane cytochrome C, caspase enzymes are activated and ultimately apoptosis is initiated. Right. But I guess there's a balance here between that, which would then be positive, shutting that cell down, versus proliferation. And might that be then the nuclear DNA modification that then allows this to propagate?
You mean the proliferation aspect?
Yes.
No, it's the fermentation.
So it's the initiation of fermentation then allows it to bypass the fact that this is a mitochondrial dysfunction.
Well, because the mitochondria, as you said, they control apoptosis. The mitochondria have controlled the kill switch in the cell. Yes. So when mitochondria to recytochrome C and initiate the apoptotic cascade, that's the result of a generally functional mitochondria. But if you have cells that have defective mitochondria, therefore the kill switch doesn't work. And the cells are now already in this fermentation pathway. And it was shown by Sonen Shinisato at Tufts University that the default state of cells is proliferation. So what's keeping the cell in a differentiated state is the function of the respiration system, which is the mitochondria. So when that becomes damaged, the cell falls back on its default state, which is proliferation.
And yet we have this mechanism whereby defective mitochondria are eliminated through autophagy or mitophagy, where they self-select for their own demise. Why does that fail? And if that's the case, what can we do to enhance autophagy or the nature's way of getting rid of those defective mitochondria?
Well, that now impacts on the concept of prevention. This is different than once the tumor has already established and is now invading and growing. And I'll talk about the nature of metastasis in a bit. But to protect, and you can't, in my mind, you can't get cancer if your mitochondria are healthy. So healthy mitochondria, if the origin of the disease is mitochondrial respiratory damage, and you do certain fasting and various certain diets that maintain the health and vitality of the body, it's very unlikely that you will get cancer. So maintaining healthy mitochondria, and as I said, therapeutic fasting reduces blood sugar, increases ketones, and facilitates autophagy for failing mitochondria. And what happens is those mitochondria that don't work properly are phagocytosed inside the cell. It's actually a lysosomal fusion. And the fuels of that mitochondria are then redistributed to the rest of the cell. So that's well and good for people who are interested in preventing cancer. I would say the vast majority of people are more interested in what are you going to do for me after I have the disease, because we know how to prevent cancer, and Warburg knew how to do that. Most cancers are almost completely prevented if you follow these kinds of things. Even if you are in fact exposed to some toxic carcinogens in the environment, if you can maintain healthy mitochondrial function, you reduce significantly the risk. Even those inherited mutations, it's another very interesting thing, like BRCA1 that you hear about, the Li-Fraumeni P53 mutations. None of those mutations are 100% penetrant. We know BRCA1 is like, what, 40% penetrant? I mean, 60% of the people that have the mutation never develop the disease. So it can't be the primary cause. It's got to be a secondary cause, because only in the patients where BRCA1 causes cancer do you see the fermentation phenotype. Same with Li-Fraumeni, the P53. There's 20% of people who have P53 mutations never get cancer. So because they never caused the irreversible damage or the damage to the respiration. So prevention is keep your mitochondrial healthy. Management is how you starve the tumor cell of its fermentable fuels. And this both can be done. Both of these processes can be done. And the autophagy process plays a much greater role in prevention or in the early stages of the tumor. Because if the tumor reaches a very aggressive state, there's very few mitochondria left in the tumor cell to resurrect. So it has to be killed outright. You cannot re-educate a tumor cell to enter back into the society of cells if its respiratory system is compromised to a certain extent.
Let me just have our viewers take a breath here and recognize that you heard a lot of similarities just now between what Dr. Seyfried was talking about and what our discussions have been over the past several years in terms of approaches for brain health in general, that we are trying to shift metabolism to a state of lower oxidative stress, of less creation of both inflammation and these damaging reactive oxygen and nitrogen species. That said, the damaging effects of the perturbed mitochondrial function in the creation then of these enhanced free radicals is a nuclear event affecting nuclear components, protein, fat, and most importantly DNA. But your contention is that cancer is not a nuclear event, but that seems to be where all the research is looking at nuclear DNA and nuclear issues.
Right, and most of it is secondary. And the strongest evidence for that are the nuclear transfer experiments, which I bundled up for the very first time. They have always been present in the literature over decades. They were just what was considered anomalies. They were not dealt seriously, and people felt very uncomfortable. They didn't want to deal with it because it wasn't consistent with the nuclear problem that cancer is a genetic disease. And this was done by a number of distinguished, the best developmental biologists that were present and still are present in the field, taking the nucleus out of a tumor cell that has the so-called driver mutations and somatic mutations and all the genetic so-called defects, and having the cell growing out of control. And you take that nucleus and now put it into a new cytoplasm, and you can develop new tissues and new organisms from the nucleus of the tumor cell that supposedly contains the origin of the disease. And what happens is usually those nuclei, those tumor nuclei, abort development, but they do not cause the cancer. They do not cause the prime phenotype.
What we've seen in Alzheimer's, in similar experiments, but with transfer of mitochondria, we do develop the Alzheimer's model in rodents that have received mitochondrial transplantation from those that are genetically predisposed to getting Alzheimer's disease. So that is exactly supportive of your contention using the Alzheimer's metaphor.
Yeah, and I think, without the NCI had a few years ago, I don't know if they still do, they had these provocative questions that were supposed to define cancer research for the rest of this century. And, one of the provocative questions is how come people with Alzheimer's disease are at lower risk for cancer in general? And what we now know about Alzheimer's disease, it's a hypometabolic disorder. It's a global energy problem. And there's fewer glucose transporters. Glucose metabolism is reduced in the brains and in the system of these. It just so happens that the brain, the neurons, are more vulnerable than other cells in the system. You're not going to be able to generate a tumor if the very structures needed to bring fermentable fuels into the cell are diminished. And that's one of the connections between the lack of the reduced risk for cancer in patients that have Alzheimer's. That was one of the provocative questions. And neurons, as I said, they shrivel up. So the neurons are suffering because they can't ferment in a consistent way. So they're going to be energy starved essentially. And this is why ketones, by the work of Richard Veitch and a number of other They're using ketone esters to prevent the degeneration of neurons in Alzheimer's disease, and this is almost the inverse of what cancer is. It's a hypometabolic rather than a hypermetabolic system.
So for our viewers, I would refer back to the interview that we did with Dr. Mary Newport,
Yes.
and also the work of Dr. Mark Mattson and Dr. Richard Veach as well that we've quoted many times. So what I think I hear you saying is, by and large, the best diet to be on is one that favors the body's production of ketones. In other words, a higher fat, lower carb diet.
I think it's good for people to do that periodically. I'm not saying, Mediterranean diets. There's so many diets that are really quite healthy for the body. But if one were to be overly concerned about their risk for cancer, suppose they have a family history or whatever, therapeutic fasting, which is just water only for several days, then transitioned into a more ketotic state, like bulletproof coffee or avocados, or what you do is you maintain the consistent low baseline glucose and keep elevating the ketones, and that increases the delta G prime of ATP hydrolysis. It's a very sophisticated but complicated system that Dr. Veach has clearly shown. You produce much fewer oxygen radicals, but you enhance the health and vitality of the mitochondria. You don't need to do this all the time. I think people sometimes fear that, the rest of my life I have to spend eating avocados and fasting or something along these lines, or eating butter or whatever. And that's not true. I think people just have to be cognizant of what they need to do periodically to restructure and regain the health of their mitochondria to prevent a variety of different diseases that are linked to energy defects.
So then a periodic fast followed by introduction of MCT oil, coconut oil, other good fats, would allow us to enhance autophagy, get rid of those defective mitochondria, really basically clean house and hit the reset button. Yes,
exactly. And that was part of chapter 19 in my book, where I called it autolytic cannibalism. It's basically a way that our bodies go after the inefficient cells, and then they phagocytize them, and they just redistribute the nutrients to the rest of the cells in our body. So we're rejuvenating, because don't forget, death is simply entropy, which is disorder. And what maintains, we can never stop this. We can just simply delay it. All right. We can either accelerate entropy or we can delay entropy. And entropy resides in the energetic efficiency of our respiratory system. As we age, our respiratory system goes down, and a lot of cells die, and we get old and we die. But that's simply the concept of the second law of thermodynamics, which is entropy. So by keeping your mitochondria and energy efficiency, you will delay entropy. That's basically what we can do. We can never prevent it, because that's the second law, but we can certainly delay it. And these therapeutic fasts and these kinds of things are simply a way to delay entropy, reduce the risk for Alzheimer's disease and a variety of these other various chronic diseases, type 2 diabetes. I mean, when you think about it, they're all related together. They all put stress on the efficiency of our energy system. Some people get cancer because the hypoxic cause of that leads to an upregulation of fermentation in the cyclic event. Other people could get Alzheimer's. Some people get type 2 diabetes. Whatever it is, it's generally the impact of a lifestyle and diets on the efficiency of our energetic system, leading to a variety of maladies, which represent probably, and you know better than me, maybe 80% of all the afflictions we have in this country are in one way or another related to one of these disturbances in energy balance.
Well, according to the World Health Organization, this is now, these types of diseases, the #1 cause of death on the planet. And, we live in a world of where people are trying to just look at everything through a very microscopic lens, of specialization when some of us, like yourself, are taking a step back and looking at the broad picture. What are the underlying common mechanisms here that seem to be at play involving things like cancer and chronic degenerative conditions, including Alzheimer's, coronary artery disease, type 2 diabetes, and even some of the other inflammatory conditions. So I think, interestingly, from a historical perspective, what you are discussing and what we've talked about is really getting our physiology back to a place where it was prior to this bombardment of our bodies with sugar and other forms of carbohydrate. Truthfully, we've never had access to sugar, certainly like we have now, and we sought out fat as a concentrated source of calories, though we didn't understand that, during our periods of hunting and gathering, oftentimes having to fast by necessity put upon us for who knows how long, days at a time. So, this is likely the condition of the human for 99.6% of our time on this planet. And, our microbiome adapted to that, our mitochondria adapted to that, and suddenly we've turned the tables in terms of food representing information, and look at what's happening around us. It's no surprise.
Yeah, and I don't think we're ever going to change that. That all we have to do is become aware of it. And I think if we know what to do, we can at least take a little bit of charge and control over the system. The food industry, it has specialized in producing foods that are unbelievably tasty, depleted in nutrition, but remarkably tasty, because we've always struggled to have things sweet. I mean, we would have to wait for seasonal ripening of berries and things like this, and then we would get very excited. But it was only temporary. Now we have it 24/7. Any supermarket will provide you with that same level of pleasure. But so we know if we can just periodically take a step back and say, okay, I'm not going to deprive myself of a big pizza every now and then, but everybody or whatever what pleases someone. But at least what you can do to occasionally reconfigure your energy metabolism.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
We published a paper showing the glucose ketone index calculator, and I was kind of surprised. It was for cancer patients, basically. But I was surprised at how many people who are interested in just general health have embraced this calculator. What it does is it measures the level of the ratio of blood glucose to blood ketones. And basically, when you are therapeutically fasting, you have a very low glucose ketone index, below 1, which means that there's more ketones circulating in your bloodstream than there is glucose on a millimolar on a regular concentration. And people experience all these, wonderful health. It's like therapeutic fasting, ketogenic diets. People look to see how their index is. And I see many of these athletes, elite athletes, and various health guys, health gurus, and all these kinds of people. They're all many of them are discussing this their index. What's their index? And it's a way of knowing how healthy you are at a particular period of time. Now, we can't stay in those health zones forever, but every now and then it's good to enter them and hang out there for a short period of time and then go back into the regular environment. But
what's a short period of time?
well, I would say that if you can get your index in the range of close to 1.0 or below, for like 3 to 4 to 5 days, I think you're doing remarkable health benefits to your body. This is the situation where you're going to now enhance the health and vitality of your mitochondria in the cells of your body. So you'll be reducing oxygen-free radicals, using autophagy to remove those cellular components that are not efficient, even removing some cells that are not efficient because they'll be phagocytosed by our immune system. But at least you have a guideline because people didn't know in the past. There was no biomarker that you could look at and say, oh, what's your index? If you have an index 1.0 or below, you know you're in a state of therapeutic ketosis. And you know that your body is now in a healing mode. And you're going to know that this and how long you want to stay in that zone is up to you. But at least you say, there's some people who checked our stuff and they did a 5-day water-only fast. And they showed that exactly as we published, they remained in these zones. And then they go beyond that. They started doing 10 days and this kind of stuff. I mean, the human is an addictive. They seem to go to extremes on certain of these. I never say, do a 25-day fast or something like that. But some people do this. And they're always trying to keep as long as they can in these zones. And and we always go overboard, in some of these things, but you don't have to. I mean, it's just, dovetail the experience in with a religious experience, a retreat. I don't know. And people can do this however way they want to do it, but they don't have to feel that they have to do this, for long periods of time. It's just very interesting. It's the human nature.
Well, let me thank you for sharing your time with us today. This has been incredibly powerful information, and I know that people who are watching this are really going to get a lot out of it. And again, this is a book for every healthcare practitioner who needs to be on your shelf. So thanks so much for being with us today.
I just want to say one more thing. Travis Christofferson has written a book called Tripping Over the Truth, The Metabolic Theory of Cancer. And it was based on my book, and it's written more for the layperson. My book is written for the not only the layperson but the scientist engaged in the field. But I think Travis's book is really exploding on the various bookstores and things like this.
Well, that's a great plug, and the name of the book is Tripping on the Truth.
Tripping Over the Truth.
Tripping Over the Truth. Okay, we'll see if we can add that into the blog that will accompany this video. We'll put a link to that. That's terrific.
Okay.
Okay. Well, we'll talk soon. Thank you again.
Okay. Thank you.
All right.
Real
Bye-bye.
Bye now.